Today we will discuss about role of IRF7 in M1 macrophages that impacts the biological function of pancreatic cancer cells. In the previous video we have discussed that IRF7 in the M1 macrophages affects the cellular signaling in the pancreatic cancer cells. How this cellular signaling gets affected, that we will cover in brief in this video. We will discuss the results that are presented here. So what they did is they first prepared a IRF7 knockout M1 macrophages using CRISPR-Cas9 technology, and simultaneously they also prepare a system where they can overexpress IRF7. So when they and then they co-culture these M1 macrophages populations with the pancreatic cancer cells, and thereafter they check the lipid numbers with the help of metabolites study, and there they have find out that in the case of IRF7 wild type, the lipid numbers are lower compared to the case of this IRF7 knockout. That is, IRF7 knockout has more lipid numbers. And similarly, when they check the lipid numbers in the experiment where they overexpress IRF7, they see the lipid number goes down, whereas in the case of wild type or uh, control, they see that lipid numbers are high compared to the overexpression of IRF7. So IRF7 overexpressions has a negative role towards this lipid metabolism of the pancreatic cancer cells. And after that, they need to check this cell viability in different conditions, and they can see that when the IRF7 is been knocked out, they see the more pancreatic cancer cells are viable compared to the case where IRF7s are knocked out, compared to the case where IRF7 is present within the M1 macrophages. So this is the two comparisons, and these are the other two comparisons where if the IRF7 is overexpressed, they can see the further decrease in the cell viability compared to the case where compared to the case of control where IRF7 is not overexpressed. This indicates that IRF7 has a negative role towards these pancreatic cancer cells viability, and also it has a negative role, or we can say that inverse role against these lipid numbers or lipid metabolites. And thereafter they check the percentage of the cells that are in which phase, and what they have find out is that in the case of this IRF7 wild type, the equal number of percentage, almost equal number of percentage are in both G0 and G1 phase and S plus G2 phase, whereas for the case of this IRF7 knockout, whereas for the case of the IRF7 knockout, they can see that there is for the knockout there is an increase in this S plus G2 M phase, and similarly for the case other two experiments, that is where the IRF7 is overexpressed, they can see that increase. They can see that increase in the number of S plus G2M phase compared to the control case. So this S plus G2M phase increase is consistent where IRF7's amounts are higher, or where IRF7 is high in the M1 macrophages. So, and these things they have validated using the flow cytometer experiment also, where they can see that this G2 plus M phase are higher in the case of IRF7 knockout, and also where this IRF7 is not overexpressed. Whereas when the IRF7s are present in the nominal amount, that is in the wild type, they can see that amount of S plus G2 phase is lower compared to the case of IRF7 knockout. And also when the IRF7 is not overexpressed, they can see that amount of S plus G2M phase is higher compared to the case where IRF7 is overexpressed. and thereafter they check this proliferation assay using this EDU dye, and they can find out that in the case of this IRF7 wild type, in the case of this IRF7 wild type, they can see the proliferations are lower, and also where this IRF7 is overexpressed, they can see the proliferation are lower compared to the case where IRF7 is been knocked out or IRF7 is not overexpressed. This comparison of these two is can be done because these two are the same experiment. Comparison of first two can be done because they are belongs to the same experiment, or same reference can be used for these two experiment. So we can see that for the wild type, the proliferations are lower compared to the knockout, and for this IRF7 overexpressed, the proliferations are lower compared to the case of where IRF7 is not overexpressed. And these things are quantified on the right hand side, where we can see that when the IRF7's amounts are lower, then they can see that high uh, proliferations in the pancreatic cancer cells. And next they did the transwell assay, and in this case also they can find that in the case of this wild type, when they compare the wild type with the knockout, they can see that proliferations are higher in the knockout compared to the wild type. And similarly, for this IRF7s where overexpressed, the proliferations are lower compared to the case where IRF7 is not overexpressed. And these things are quantified on the right hand side, where they can see that when the IRF amount are lower, then their migrations or invasions properties are invasion properties of the pancreatic cancer cells are higher. And thereafter they did this staining assay also. This staining migrations checking also, where they used this IRF7, where they check it for this IRF7 and wild type and knockout, and they can see that for the knockout the migration, and in the panel H and I, they did the experiment for in order to check the migrations property of the pancreatic cancer cells in the uh, presence of macrophages with IRF1 and without IRF1. So without IRF, when the IRF1 is knocked out in the macrophages, they see that more pancreatic cancer cells are migrating compared to the case where IRF7s are present. And similar results are obtained where IRF7 is overexpressed, shows low migrations compared to the case where IRF7 is not overexpressed. And these things are quantified on the right hand side, where we can see that similar quantifications, that where the IRF7's amounts are lower, and they can see that more migrations. And similarly for the invasions experiments also, we can see that when in the case of IRF7 knockout, there is more invasions compared to the case of IRF7 wild type, and similar results are obtained where we can, for this overexpression of IRF7 shows low invasions compared to the case of this IRF7s are uh, IRF7s are not overexpressed, and these things are shows the similar results. Quantifications are shown on the right hand side. Next they check the apoptosis. So apoptosis also results are also in line with their other results, that in the case of wild type they see that in the case of wild type they see the low apoptosis, whereas in the case of this in the case of wild type they see the more apoptosis compared to the case of this IRF7 knockout, where they see the low apoptosis. And these things are also in line with the other studies where we can see that more migration properties, proliferation properties, invasion properties, and also the lipid metabolisms are higher. And similar results are shown for this, where IRF7 is overexpressed, they see that more more apoptosis is more compared to the case where IRF7 is not overexpressed, so indicating that IRF7s as more the IRF7 will cause the reduction in the pancreatic cancer cells, and this increase in the apoptosis could also increase or the increase in the cells in the G2 and M populations could also be because of the low apoptosis of the cells in this IRF7 knockout, and this IRF7s where is not overexpressed. So all this in conclusions, what this experimental results suggest that IRF7s is inversely proportional to this pancreatic cancer cells progression. So if you need to understand more about the experimental details, how they have conducted their experiments, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.